our YouTube channel. So thank you, folks, for joining us for our honors colloquium here in early April. We are very pleased to be joined by Jerrod Tynes, who you will see maybe after the presentation when we go back to the camera, who's an adjunct professor in our biology program here at A&M Texarkana. And I think has done one of these for us before.
Never done.
Oh, have you not? I thought you did one of these before. Well, never mind. First time for everything. Today, we're going to be hearing about From Snakes to Scrubs, herpetology research within the field of medicine. We'll have some time at the end for questions, I think. And otherwise, Jerrod, take it away.
Thank you. So thank you guys for joining me today. So yeah, I am a biology adjunct here. I teach actually only online. I teach animal nutrition typically in the fall and usually herpetology in the spring online. I've been with Texarkana since 2018, and this is actually my first visit to campus. So very much an online professor. Full-time, I am a assistant professor for a Master's of Physicians Assistant program in Richardson at West Coast University, Texas, just north of the DFW area. And then I also teach genetics online for at Dallas College in the DFW area. So just to kind of give you an overview, I'm just going to briefly go over some of the research that I've done and kind of how that's led to my full-time work and what I do specifically within the realm of higher education and research and integrating and intertwining the medical work that we do. Like I said, I work for a PA program, and I teach the anatomy physiology class for the physician's assistant program, and I also teach the capstone coursework and the evidence-based medicine course. And so from early on, I've been interested in research specifically associated with reptiles. I grew up on the lake just outside of Dallas and Rockwall. And so I was always catching things. I was always interested in that. And I was as an undergrad, I was a member of the inaugural class of the Honors College at A&M Commerce. And so very familiar with undergrad honors research. I worked with Professor Lanny Lyman Henley. We did behavioral work. And so what we looked at and what my undergrad thesis was I analyzed and evaluated climate behavior in the North American rat snake complex. And so for those of you that don't know, there's a large variety of species of North American rat snakes found in Texas, as well as the southern United States. And so I was at Commerce from January 08, graduated my bachelor's in 11, and then stayed for graduate school there, where I was a graduate teaching assistant and research assistant for a couple of different professors, right? So here's what I did in my undergrad and graduate work. I basically set up climbing tanks. Now, from a behavioral standpoint, rat snakes are typically in documentation are considered to be arboreal. However, most of that research is done with wild specimens, with animals that are out in the wild. And so very little data was collected on or had been collected on captive species. And so that's what I was really, really interested in, taking a look at if they didn't have any previous exposure to capturing food, which is a main reason we see wild rat snakes climb out in the field. I had captive bred, captive fed specimens. And so I wanted to see, did they have an innate behavior to climb? And that's really what I was looking at. And so what I found is they did. They preferred to be in the upper locations, in those high boxes that you saw on the last slide, statistically significant. I also looked at various groups. So we had integrated rat snakes, and so rat snakes that are a mixture of a couple of different species, as well as pure line rat snakes, so a single species. Rat snakes and snakes in general are actually quite unique in that they can cross the genus line. And so you can actually produce viable offspring, babies that can have babies, by taking two different genuses of snake, right, from a biological standpoint and breeding them, as long as they have the same chromosome number. Rat snakes have the same chromosome number as things like king snakes, corn snakes, pine snakes, have 36 chromosomes, right? So I was also looking at the difference between males and females. We can see that we obviously both sexes preferred the top hide And do you need something?
I was going to let the person in who just came into Zoom. Sorry.
Oh, sorry.
No, you're fine. It's just the, sorry. There we go. All right. Apologies. Continue.
Right. And so from that undergrad work, and then I continued that work. The snakes that I used for my undergrad work, I actually bred them. They became parents, and then I used their offspring for my graduate work while I was at A&M Commerce, doing some components with primarily climbing behavior, but I did a little bit of other work with some, we had axolotl salamanders, which are a regenerative species of salamander. They have the ability to regenerate full limbs. So from a tissue standpoint, it's very, very unique. They're a great specimen. There's actually more of those particular salamander species or specimens in captivity than they are in the wild. They're from central Mexico and a couple of different river basins down there. Because of pollution, they're relatively endangered. And so from our finding from this work, right, we took that and looked at the ecological importance. Obviously, if we talk about species that can cross the genus line, taxonomic importance associated with behavior and genetics, which is what I did with my master's while I was at Commerce. And then we'll talk a little bit more about some pet product and pet sales work. Also, while I was at Commerce, right, I did some work in the field. And so I was capturing this beautiful specimen. This is a speckled king snake from, you guys have them around here. We have them also at Commerce, but I was also capturing venomous species. And so I had multiple water moccasins, cottonmouths, right, as well as did a little bit of work with, we had a couple of copperheads as well. So what we're doing, we had this beautiful survey site, actually a couple of different sites, artificial wetland and an artificial bottomland or created bottomland. We set up what's called herperays, so they're basically Y-shaped structures. And then along this sheet metal fencing, we have funnel traps, which allow the snakes or lizards or turtles, or in some cases mammals. I even caught a shrew in one of them, which is a very unique capture. And anything that kind of rolled into that. And then we took some data, primarily size. We were doing just analysis of the species diversity that was there. This is just a photo that shows obviously the visual differences between our two cottonmouth images here, right? And our plain-bellied water snake here and our Norty rum for our diamondback water snake on this side. I was also leading the undergrad field methods class during this time, and so we were very careful about making sure we weren't handling the venomous. We had to be very careful about where we walked. I had to know exactly what hospital to go to if we had somebody bitten, right? So all of those components. So just looking at some raw data, we can see the variety of species that we caught, and then kind of this is the difference in the arrays that they were found. We also had ribbon snakes, brown snakes, steckered snakes, right? So looking at just the overall data, right, we can see in the two sites or a couple of other sites that I was doing, had pull in some historical data, large number of water moccasins, western cottonmouth, and then some huge variety of snake species. So both in the bottomland and our wetlands. So I did both the 2012 and 2013 surveys for my graduate projects. The other were pre my time, right? But good variety of species that we were catching. It was a lot of fun. Field work is a lot of fun. It's dirty, but it's really, really enjoyable if you like being outside. This is just a continued list. Once again, we found a huge variety of reptiles when I was at Commerce. And then also some really cute things, right? So we found box turtles. We found these are baby red-eared sliders. On my walk into campus, I saw that you guys had a couple of musk turtles at the pond down the hill, right? So, and then we also did mark recapture. And so for turtles, what that looks like is we would basically take nail polish and we would paint the underbelly of one of the scutes of the turtle. I would take a photo of it and then I would record that. And then if I caught it within the survey, it was in a 1 month time frame. And so if I caught it again, that nail polish would still be visible, right? So that was how we would mark recapture. The other specimens, we would simply denote. So wild reptiles look rough compared to like the pretty ones that you see in the pet store. They are beat up, they have parasites, they have cuts, right? They just look beat up. And so you can take enough photos of them to really, for the most part, be able to reevaluate if you capture the same lizard or the same snake. And then some of the frogs that we found, just like this bullfrog here, huge variety. The eastern narrowmouth toad was actually a really good find as well. And then just some data associated with that capture. There's just some numbers there. Huge number of leopard frog, which is the photo here on the left. We had lots of those captured. And also, we were able to catch a couple of these guys. This is the Texas garter snake. Now, we weren't doing work with these guys at the time in commerce, but Dr. Neil Ford and Dr. Playchick and their grad students out at UT Tyler was doing some work with them. And so we were able to send them some samples and some information associated with the Texas garter snake that we had. I've also had the opportunity to work at the Dallas Zoo. So I've been an education instructor for the Dallas Zoo for about 9 years, really pre-COVID, and then COVID hit, and then the world shut down, including they dismantled most of the education department at the zoo. And so my wife and I were both on that list, right? But while I was there, we did a lot of really cool stuff. Primarily, we did Boy Scout and Girl Scout groups. We would do VIP groups behind the scenes. So I've been able to see really behind the scenes of a lot of the places at the Dallas Zoo. We did special events when VIPs would come in. And some of the work that I did there is I was specifically looking at and interested in kind of what the reptile industry and the culture, herpetoculture, so keeping reptiles in captivity, what kind of potential there are for problems, specifically with things like release. And so just some basic history of pets in the industry. We live in a state where reptiles are readily available, not only outside, but you can actually purchase them. I can go to Walmart down the road, right? Pay for a $15, it's probably $20 now, Texas Parks and Wildlife Class 1 exotics permit, and then I can go buy a $1,000 rattlesnake at a reptile show if I want to, and have it legally, right? And so here in Texas, we have one of the loosest kind of set of wildlife regulation laws, which, you know, a few months back, there's that individual down in Houston that had the tiger that got out, right? You're always hearing about different things. So that is if you are interested in keeping exotics, that's a kind of a good thing. It's why we attract it, but it's also kind of a bad thing, right? In terms of if you're not handling or don't know what you're doing with some of these species, right? So we have a huge variety, as I said, of Texas species of snakes. And a lot of these are found, you go to a reptile show, you go to a pet store, and you're going to be able to potentially buy the same species that you might be able to catch outside in captivity. Now, in captivity, obviously, most of these are captive born, and so their parents and grandparents of these snakes and lizards have been raised in captivity. And obviously, because of that, we also have some specific mutations that you're going to see in the reptile industry, like albino or axanthic, right? Something a genetic mutant that probably would not survive in the wild, right? So these are just some of the species that are sold on a regular basis. Anywhere between, you can pick up king snakes between 30 and 40 bucks at sometimes the reptile shows. And then just assuming there's a specific morphs associated with these, most expensive thing on this is probably something venomous. So rattlesnakes are between $300 to $1,000 typically if you're purchasing those. And then hognose snakes, because of kind of the interest in hognose within the last couple of years, those are $500, $1,000, $2,000 depending on the mutation. Right, and actually, this is some photos from that reptile or a reptile show that I not only talk at, but also occasionally did. My wife and I do breed reptiles occasionally, right? So we've got $250 for this Transpecos. These are $65 for the baby Baird's rat snake, $135 for the Transpecos, $350 for this hognose. That's a relatively cheap hognose for one that size. And then I think this says $750 for this anaconda, full-size breeding hognose, right? So So why is it bad? And there's certainly nothing wrong. I'm a pet owner of multiple reptiles that had literally everything that you can pretty much think of. In terms of the reptiles, I've also had some crazy things, like my wife and I were hedgehogs for a few years, which was a lot of fun. But I've had caimans, right? I've had a lot of different things. And so one thing, especially for snakes, they like to get out. They like to escape. They're very, very good at escaping. So snakes and really tarantulas are probably the two kind of groups that are the best at escaping. They're just amazing. They're just amazing at getting out of cages, right? So you have to, in terms of pet ownership, that's super important. And then in some cases, snakes are released. So this bottom photo is a Dumeral's boa that was found, I think, in Bonham, Texas, right? So the area outside of Commerce region. A student actually brought it in to me and Professor Henley, and it was a 6-foot boa, Dumeral's boa. These guys are from Madagascar. They eat lemurs on the island of Madagascar, right? So very dominant predator. And in fact, in the island of Madagascar, it's the top of the apex predator of that island, right? And so they're not really going to survive out in Northeast Texas, and so it was dumped. It was an animal that was just probably released. It might have gotten out, right? But there are occasional ones that are dumped. And this is a photo of an albino, once again, something that we're not going to see in the wild, albino corn snake that was found just east of in McKinney, right? Just east of the Herpetology in McKinney. And then if we're talking about some other kind of problematic issues, is we can potentially see the variety of things get out, like if we had a captive-born king snake get out and just genetically potentially cause problems within its native range, right? So we have to be very careful about those species that have the potential to get out and potentially interbreed with some of the wild stock that we have out, right? And so educational opportunities. There's a lot of great things that we can do, right, with reptiles in the classroom, right, in the research facility. As I've talked about, I've done field work, I've done behavioral work with them. When I was a high school teacher, I had probably 15 or 20 in my classroom. When I was at A&M Commerce, we had our research lab that had about 300 snakes, or 200 to 300 snakes, right? So there's a lot of interesting things and we use them a lot of times on programs at the zoo. We would have specific snakes that we would bring out, let the kids touch and hold, or not hold, but touch as an educational opportunity. From the zoo and after Commerce, I went and pursued a second master's, specifically in pedagogy and education teaching. And so from there, I was doing cardiovascular work with common snapping turtles and American alligators, right? And so some of that work, we basically would keep turtles, right, or alligators in large scale tubs like this. We would get eggs, raise eggs in hypoxic, normoxic, or hypercapnic conditions, so high CO2, normal O2, or low O2. And then we would look at the cardiovascular plasticity, or the alterations of the heart during egg development when they turn 3 years old. So we actually had to hatch them. Baby alligators, the cutest little things, right? They're like this big and they cry. They're a lot of fun to work with. They still bite when they're little, right? We would hatch the eggs out. We would raise them in these tubs for 3 years. And then we would do cardiovascular surgery on microsurgeries on these animals, which was a lot of fun. Surgery is not a lot of fun in about an 80-degree temperature room you're talking about 4 or 5 hour surgery, right? So typically when you think of surgeries, like when we have surgeries, right, the rooms are usually very, very cold. I work with staff who came from clinic, right? And then when he did ortho surgeries, he said that the room is typically about 55 or 60 degrees for surgeons and all the medical staff that are within that, right? But we're mammals. These guys are reptiles. They can't maintain their body temperature. And so in order to do the surgery with them under drugs, we had to keep the room at the appropriate temperature, which was problematic for us. It was very, very, you were like sweating into the animal. They were also slightly cannibalistic. So typically in the wild, they were, yes, what is slightly? So if you look at kind of ecological data or what they would feed on in the wild, they're not necessarily cannibalistic, right? So now you keep them in a tub that's this big, and you've got 3, 4, 5 apex predators in a tub that's big. So we had to be very diligent about keeping an eye on them. We would clean these tanks every single day. We'd drop in basically a pond filter, suck out all the water, fill it with water every single day. Go to grad school, right? That's basically like if you're working with animals in grad school, that's a lot of what you'll be doing is cleaning, right? So if you want to go to the zoo, that's a lot of what you'll be doing is cleaning. But it was enjoyable. There were times where I would get a 3 a.m. phone call and an alligator gotten out, and the custodian walking through the lab was like, "There's an alligator out. You need to come get it." I was the grad student that was on call that night, and so I would have to leave my apartment, go figure out. Because we're talking like 200 alligators and about another room of 200 snapping turtles. So when they got out, you had to ID them and put them right back in the appropriate, tank, right? So, and then especially the snapping turtles, it was not uncommon for us to come in, and one of them had basically started to chew off the limb of another, and they'll basically just start at a leg and go all the way through until they hit shell. Right, so we would have to come in. Sometimes we'd have to euthanize the one that was attacked. Sometimes we could suture it up and it would be okay. Now, at that point, it's no longer a kind of a viable specimen for research, which was not a good day because my professor was very unhappy when that happened. So this was an NSF, a multi-million-dollar NSF project that we were working on. Dan Cross is a NSF careeree, so he's had millions of dollars over his career time. And this was at UNT, right? So we didn't do the breeding. We got the eggs from specific facilities that did the breeding for us, but you can breed these guys. I did a couple of talks at like reptile conferences on how to breed snapping turtles, but you do see occasional mutations in turtles like the albino one up top, but rare. Right, and then once again, what we were looking at cardiovascular mechanics of diving. So we would put them on treadmills, or we put them in dive tanks, and then look at cardiovascular. We'd read EKGs from their cardiac surgeries. We would basically go in, put aortic clamps in and around their heart, and then sew them up, and then put them in the dive tank or put them in the treadmill, and then run and look at EKG. And once again, we were looking at cardiovascular output. And based on what clutch they came from, were they hypercapnic, or they hypoxic, one of those. And then urban wildlife management, looking at, we have, you guys have alligators here, right? And they're native to this far east of, we have them as west as Fort Worth. There's a couple spotted in Lake Worth not too long ago, right? So most of the specimens that we got were from Louisiana. And then once again, right, we did find that heart physiology is highly plastic. So when we talk about plasticity in biology, that actually means that it's moldable, right? It's kind of counterintuitive to what we think about. But we did see there's significant differences based on their environmental conditions that they were raised in. During that time frame, after I was done with my second master's, I took a job as an EP environmental science teacher at Forney High School. I stayed in academia as an adjunct and worked with my mom, who is a professor of nursing at Louise Harrington School of Nursing for Baylor University in Dallas. And so specifically, we wanted to look at pathology and some components of venom in nursing care victims. So she was actually planning a Baylor trip with some students to go to Vietnam, I think at the time, and do some nursing care and nursing training. Baylor they send a lot of their faculties out to international nursing schools, and then they do international training, both with faculty there as well as students there. And so they were headed out to Vietnam. There's a lot of different venomous species in Vietnam and Southeast Asia. And so we were looking at kind of honing some techniques and some teaching techniques to teach to nursing students about how to deal with a snake bite victim, and then make that transmissible. Now, that's not necessarily a thing that a normal nurse, even an ER nurse in the United States, there's only about 7,000 to 8,000 snake bites annually in the US, the CDC reports. Only typically about 10 of those are fatal. Usually those are males between 18 and 34, and about 80% of those are involved with alcohol, right? So as a nurse in the United States, there's not a whole lot, and I teach this to my PA students, that you're not going to really see a whole lot of snake bite victims. You may see a few. I work with a colleague who's our pharmacist. She teaches our pharmacotherapeutics course for our PA students. And she'll tell me every time someone comes in that's a snake bite victim and kind of what anti-venom they use or if they chose to. But it's just not something that you're going to see. Here in the US, right, our three main types of vipers that we have are listed here. We have our copperhead, which you guys have in this area, water moccasins, which I'm sure there's a few down in the pond, and rattlesnakes. Now, if you guys don't have this, but this is a rock rattlesnake from Central Texas. Y'all have cane bricks probably and timbers here. Right, and crotalid venom is considered hemotoxic. Now, it's got a whole bunch of other toxins, and so part of the work that my mom and I did was an extensive kind of meta-analysis, a lit review of different types of toxins as well as techniques to teach nurses, right? But just kind of an overview of some of the toxins, right? There's neurotoxin, cardiotoxin, myotoxins. But hemotoxin and myotoxin is typically what we think of when we think of North American vipers. So vessels erupt, muscle tissue starts to degrade. I will let you know the next couple of slides are not pretty slides. There's some nasty tissue images. So if you want to close, I'm just letting you know, right? So like that. So this individual, right, had been bitten on the hand, and I cut some of the image off. But basically, his arm has exploded from the swelling from this hemotoxic venom. This guy was bit by a rattlesnake, right? So looking at things, so obviously shut down hematological cardiac. We can go into cardiac arrest. I did some work. I had a colleague that I met at a toxicology conference presenting actually this work and some similar work, who was one of the chief pathologists for Oklahoma City. And he had a snake bite victim, at least that's what he thought. But the individual had snake bite marks on his leg, but there was absolutely no tissue swelling whatsoever. So he called me, was like, hey, like this is what I have. I'll send you the case file over. And so I looked at the case file and I was like, well, it's not super common, but occasionally an individual can go into shock or go into like immediate shock and the individual die from anaphylaxis very, very quickly before actually there's any tissue damage whatsoever. And so that's actually what he presented at trial for the COD, right? So, but typically we see just like this, so tissue edema, reddening, swelling to the point where often from a emergency standpoint, you have to drain the tissue or the physician will cut it open just so it doesn't explode, it doesn't rip the tissue. They'll actually go ahead and make a surgical cut to allow the swelling to kind of expand. From a nursing assessment, well, one thing we typically do is we do a bite analysis where we take timestamps of swelling. And so here in the image, it's kind of fuzzy, but we have the snake bite here, and then we can timestamp the swelling. So nurse will come in, 30 minutes, right? Circle where that swelling is, like 45 minutes or an hour or whatever it is. They'll continue to do that to kind of trace the swelling. But obviously, most important is ABCs. I think they've changed that acronym since then. But airway, breathing, and circulation is what we're really looking for. And then we really do need to know from a comprehensive medical standpoint, right? Is the patient diabetic, right? Do they have a previous history of cardiovascular problems? Are they on medication, right? All of those things are extremely important in terms of understanding what's the appropriate course of action, especially if we're going to do something like give anti-venom. And so relevance. So once again, in the United States, snake bites are really not that big of an issue. In other countries, they are. If we look at something like Nepal, 162 right, per 100,000 died snake bites. In Nigeria, it's higher than that, right? The 12.2% mortality rate, which is relatively high. And we're talking about a lot of these regions, right? I'm sure there's large volumes of unreported data, right? Villages where there's just not information that gets out for us to actually make these statements. And then once again, in terms of you can buy, this is at a reptile show. Every single red tape means that's a venomous species of snake that's being sold right there. Right? So once again, in Texas, we have a very kind of, I would like to say, loose control over exotic species. And so I can go buy a eyelash viper at a reptile show for $250, right? So now, the biggest issue with some of these exotic species is if I am keeping an eyelash viper or a gaboon viper, your local hospital is not going to have anti-venom for that. They're going to have anti-venom on site for the vipers that we have in the United States, which is Crofab A or Crofab B. There's a new venom, which we'll talk about in just a couple of seconds. But they're going to have that. They're not going to have the exotic anti-venoms. And so if you're bitten by an exotic species, a lot of times what hospitals will have to do is they'll have to call zoos and get anti-venom from zoos, because those are the only places that are going to have like polyvalent or covalent or elapid anti-venoms, right? So luckily, if you're bitten in kind of Northeast Texas area, Dallas Zoo is has the largest number of venomous species in the United States, the largest collection of venomous snakes. It's also the largest breeder of venomous snakes, which means they have 2 or 3 refrigerators of anti-venom on site. So it's not uncommon for us at the Dallas Zoo to get a phone call and say, hey, yes, we have Cape Viper venom, anti-venom for you. So come pick it up. Right. So from there, I went to UNT Dallas. I started an urban agriculture renewable resources program there. And so I specifically did a lot of research. I ran an undergrad research program. I also oversaw a capstone project course where I would put students kind of in internship-based courses. But from a data standpoint, I was really looking at adding certifications integrated within curriculum for college students. So when they graduate, they not only had their degree, but they had industry-based certificates, which is really, really important, especially in agriculture. That's really why we were focusing on it, because if you look at something like pest management or nursery landscape or welding or things like that, you have to have additional certifications. And really, at the time at UT Dallas, we had a biology degree, a BS in bio and a BA in bio, and we were struggling to see students get industry-based entry-level college or entry-level positions associated with their degree, right? And so we wanted to look at and evaluate how we can make those students more competitive. Both in my kind of urban ag program, we started a BAS in urban ag, as well as a lot of my students that took my classes were also biology students. So I looked at working with those students as well. So this is one of the community gardens that we started in the area. So this institution is located in what we call a food desert and also a food swamp. And so a food desert is a place where there's not a large volume of healthy nutrition for the population, right? So this institution is in South Dallas at near Oak Cliff area. And then a food swamp is where there's large concentrations of food, but it's all bad, right? It's all like fast food or food that you get from gas stations, right? And so like this place was this institution, and the students that we served, it's a Hispanic serving institute. We had about, I think it's 78% Hispanic population at our institution. And so we were looking at a lot of nutrition and food swamp and food desert components, right? But shifting back to our analysis of the certifications that we were doing, once again, I integrated certifications within my coursework and then took a couple of different types of feedback from students. So these are just some statements from my faculty evaluations, but this was important to me, right? So the students thinking that and hoping that, you know, their certification or their digital credential was going to get them into their future career, right? So, looking at some of that survey data, right? This is the number that were in these courses, and then also some of the ones that were recommending the course, completed the industry certification. So our goal was to obviously get this number up, right? But we can see some of the certifications. And I was working with a huge variety of organizations. So, for example, I worked with TNLA, which is the Texas Nursery Landscape Association. I taught a course that was a special topics course, but I took their educator prep content from that organization, that industry organization, integrated into my course, so my students could then sit for the TNLA certification exam after they were done with my course. And that was kind of what we were doing. And then I presented this at a couple of different conferences, looking at kind of what we can do as faculty, right? Find out, I always, at every course I teach, I try to get an idea of what my students want to do career-wise, especially since I teach such a wide variety of courses, and a lot of my courses in the biology department are upper level elective. And so I've got students that want to go to med school, or they want to go be a zookeeper, or want to, like, and everything in between, right? And so I try to get a pulse on how I can help students figure out what they want to do. All right, and then from there, so that was a grant-funded position, and I kind of knew going in that it was a grant-funded position, and I didn't know what my end after that grant position was going to be. And so I did what a lot of people do, is I got another education degree. So I got a certification in educational leadership, I continued my education. And so UNT Dallas, while I was there, had a principal certification program, and the state changed the requirement years in Texas from 3 years to 2. I taught 2 years at Forney. So I was qualified to go and get my principal certification after I took the 5 or 6 classes that I needed from UT Dallas. And so I did that. And I'm glad I did, because I really, really enjoyed, I was only at Jose Jome Elementary for a year, but I enjoyed my time as assistant principal and some of the work that I was able to do there. And so specifically, while I was there and finishing my certification, I wrote an 80-page capstone on utilizing kind of reptiles in classrooms, as well as looking at teaching teachers how to teach science. So we were an F campus as an elementary school. When I left, we were a B campus in a year. So this was right after COVID, right? We had transitioned from remote to face-to-face learning. A lot of schools in the state were significantly suffering, and we're seeing still some problems associated with gaps in learning, right? And so specifically, that's what we were looking at, is these gaps in learning. A lot of times too, when we talk about science teachers at the elementary level, they don't have a science degree. They have a pedagogy degree, or they have an interdisciplinary science or an interdisciplinary degree where they have a lot of courses and a lot of broad topics for their certification. And so we're looking at how we can, from an administrative standpoint, help teachers. And so we did a couple of different professional developments. Specifically, we did this, was called a Freyer model, utilizing reptiles as the example. I was also breeding reptiles at the school. We had a science lab, and so they would go, basically my snakes would tour from one classroom to another and transition. And then this is what we want to see from an administrative standpoint of our teachers, right? I'm leaving with a new resource, and I have a better understanding of the model or the technique that I looked at and evaluated from professional development. That's all obviously what we want to do. And then from a student data standpoint, right, this was important for us that as these are assessments. So as our assessments went down, or through the time frame, our masters, which is the high achievement, went up, right? So as we were working on teaching our teachers, our process was going up, right? And then I was able to present this at a couple of different conferences. And then now, right? So all of that kind of accumulated into, I left Jose Jome and took an awesome position as a full-time faculty at West Coast University, Texas. And so previously West Coast University, Dallas, when the old building was across from the medical district in South Dallas, but they changed and we moved to Richardson. And right now I'm doing a couple of works. I'm doing bacteria and substrate analysis, as well as looking at some emergency room use and anti-venom. And then I am the committee chair for our, I never get this right, we call it the P committee, but it's Policy Evaluation Assessment Committee for our program, for our accreditation purposes, right? And so what am I doing for the substrate? Well, what we're looking at is if you offer substrate to different species of reptiles, is there a change in the bacteria that we find on and in the substrate post, number of months or weeks in terms of time frame? And so that's what we're really looking at. I'm actually working with these companies and a couple others. They send me a whole bunch of basically bedding, and we try them out, and we try to do bacterial counting. And so that's what we're looking at. And we want to know, especially right from a PA standpoint, are they zoonotic? Are they potentially pathogenic? Right. So we know we're going to see salmonella, we're going to see E. coli, but are there strains that are potentially hazardous to people? Right. And so when we talk about bedding with reptiles, we have what's called the ecological option, which is the option that where you just try to mirror what they would see in the wild. Right. So the bedding there, or the sphagnum moss for a viper boa here. Or artificial substrate, right? So this is literally shredded paper for it's a hybrid king snake corn snake or king snake milk snake, or this is basically a cut of astroturf for this. That's a reticulated python, all right? So now this example was at a show, so it more than likely doesn't live on that. It was just gonna see, and then we wanted to see, especially because a lot of individuals, especially if you have large numbers of snakes, you feed them in their tank. Not a great suggestion. I wouldn't recommend it, but you can do it. And is that we're also looking at bacteria in the oral cavity and the cloaca as well to see if there's any connection. Yes?
I was going to ask, why don't you recommend feeding?
So a couple of reasons. Primarily, and it really depends on the species. One is that's a frozen thawed mouse there. And look at the aspen around it, right? So what are the chances of one of those aspens sticking and then getting in the GI tract of that animal? Pretty good, actually, right? So that's one issue. Another issue, not so much with this particular species, but if we go back to like the reticulated python, there's a lot of data that shows if you feed them, they associate basically opening the cage with lunch, right? It's very much the dinosaur from Jurassic World, like, oh, that drop means the cow is getting dropped, right? So it's the same kind of concept. And so that's another thing, especially if you want an animal to hold, they need to, they're not bright, right? They're reptiles, right? So they need to understand that opening up the top of the lid is not necessarily lunch every single time, right? So those are the primary reasons. Now I say that this is my snake, my brother's keeping, this is my gray banded king snake right here. And he's feeding a lot very, very quickly. So looking at emergency room venom, I talked a little bit about this right earlier in that typically when we see anti-venom, Profab A and Profab B are our primary anti-venoms, and really the ones that we've been using for decades. And anti-venom has been virtually the same for about 100 years. And prior to the last 2021, 2022. We're now using recombinant anti-venom, which is new. This is actually an April 22 publication, so relatively recent, where we're basically creating associated therapeutics for anti-venom within the lab, rather than utilizing an anti-venom serum that is extracted from a equine. That's what's in the example here. We also have porcine anti-venom and sheep and goat are occasionally used as well. And so, like I said, I'm looking at some things specifically, potentially some cost analysis from a healthcare administrative standpoint of these different types of anti-venoms. And then, like I said, I do a lot of PA data, right? So I'm often on my computer, I'm mining data for our program. We're a brand new program, and we're only on our second cohort, right? So we started in January of 2022. And so as a provisionally accredited program, and so because of that, we have what's called a self-study, where we have to evaluate how successful we are and how successful our students are. And so I do lots. Now, this particular survey was pulled from the PAEA, which is the Physician's Assistant Education Association. Actually, no longer, it's now PA because Physician's Assistant, and there's also programs that call those same, basically, degrees Physician's Associates. So there's kind of a debate between the same practice, right? So the world is kind of going to just calling them PAs. But I thought some of this data was interesting. I'm trying to think why I pulled this particular subset. Our students were very interested in burnout. So we're studying burnout right now, not only in faculty and in healthcare, but also in student populations. And so that's something that we're really, really interested in. I have a colleague that's working on that. And then we're also evaluating potentially increasing nutrition as a component of what we study.
We probably should call it the class.
Oh, gotcha. Okay.
Sorry.
No, that's fine. So, like I said, I'm a faculty. If you want to take my class in nutrition, there's a plug for registration. And then if you want to know more about PA programs or any of the programs that we have at West Coast, there's my information.
Excellent. Well, that was wonderful. Thank you, Jerrod. Appreciate it. And let me go ahead and end that. If we do have any questions, hold on, I can take them in the hole. We already stopped sharing. Hold on, excellent. So yeah, if you do have any questions, feel free to send us an email at honors@tmuc.edu, and I can pass them along. But otherwise, thank you very much. We will now turn it over to psychology, I think. Is that who's in here at 1? Okay. Well, thank you very much, folks.
You're an honors